joining. We'll give everyone just a few minutes to join. And just a reminder that we will be recording it and posting it. This will be posted by the end of the week. Also, for all you, in case you can't join for the whole time, don't worry about it. Come back and see what you missed going forward. Just to let everyone know, there will also be an option to put in questions into the Q&A box. I will put up a slide in a minute to show it to you, but feel free to drop any questions that you have in that box. These will be more general questions, especially if you can't hear me or see me or have a question about what's going on, and we will try to answer it. We are not able to provide like specific medical advice, and this is, as I mentioned, going to be recorded, so you may want to refrain from sharing very detailed personal health information. So we'll keep it a little bit more general basics genetics, but feel free to email us if there are additional questions. So thank you all again for joining. My name is Caitlin. Tonight we're going to talk a little bit about the importance of individuals sharing health and genomic data. This live webinar will be recorded, so then it won't be and on our YouTube channel or YouTube playlist, so we'll be able to check it out later, send it to friends, or if you miss anything, circle back around. So as I mentioned, my name is Caitlin. I'm a genetic counselor by training, and I am one of the individuals that helps support GenomeConnect, and really appreciate everyone who has joined, all our participants, and I really am excited to talk with you all tonight. And I'm going to have my colleagues who are here with us go ahead and introduce themselves and tell themselves a little bit about who they are and what they do with GenomeConnect.
Hi all, my name is Joel Savat. I'm a genetic counselor by training as well, and I support the GenomeConnect registry along with Caitlin and Inika. I'm Caitlin's tech backup today, so I'll be going off camera in a moment, but just really excited that you all are joining us.
Hello, everyone. My name is Inika Cordova, and I am one of the genetic counselor assistants that supports the GenomeConnect patient registry. I am another one of the tech backups, so I will also be going off camera very soon.
Thank you both. Thank you for your help. This next slide is just a reminder of GenomeConnect's upcoming webinar. So thank you all for joining us today at 7 p.m. We will have another evening webinar in April to celebrate DNA Day, and that is how to read a genetic testing report. That will be hosted by Alexis Morgan, who's another genetic counselor on our team. And then we have webinar coming up later on in the year on June 25th. This will be at 3 p.m. Eastern time, and this will be hosted by Dr. Maya Chopra, and talking a little bit more about the Brain Gene Registry, and that's a GenomeConnect registry partner to hear all about that one. Again, all of the webinars will be recorded and available to view later on. We did receive some questions beforehand, so thank you all. I hope that we'll address some of them, but obviously questions always come up. Feel free to put them in the Q&A box. That should be on your screen. I highlighted it in blue what it should look like, and this will be available throughout the webinar to ask those questions. Like I said before, we are recording this webinar and we are not able to answer specific medical questions, so they're going to be a little bit more generic, and just for your own protection of your health information, I would limit the amount of personal health information that's shared in the Q&A. So this is an overall agenda of what we're going to do today. We're going to talk a little bit about what is genetic data, and then I'm going to move on to how individuals can participate in data sharing, why data sharing is important, what are the benefits to the individuals that participate, and then we'll go through and open it up to questions and answers. All right, the first question is, what is genetic data? So each of us has more than 1 trillion cells that make up our bodies. We have different types of cells, including our skin cells, our brain cells, our heart cells, and these are all the building blocks of our body. In each of our cells, all of our genetic material or our DNA can be found. Our DNA is like our body's instructional manual. Each instruction is called a gene, And to keep our DNA organized in our cells, it's wrapped up tightly in these structures called chromosomes. I like to start with this background to get everyone on the same page about what we will be talking about in terms of genetic data, and that really is involving in the information that are in our genes. So we have many changes in our DNA, and these are called variants. So these are changes in our DNA, changes in our gene. Some variants can impact how proteins work in our body, and some of these changes can cause developmental differences or health differences. And so these are what we're going to be focusing on kind of for the whole talk when we refer to genetic data or genomic data, these genetic variants. Okay, so those are genetic variants. And now how do we identify genetic variants? And this is a little bit of the types of testing that's available in order for us to learn about these genetic variants. So the first testing type I want to mention is karyotype. A karyotype is a test that is used quite frequently, and it looks at chromosomes for large copy number variants. So looking to see if there's a whole missing or extra chromosome, if there are large pieces of chromosomes that are missing or duplicated extra. And then we have another test called a microarray. And this is if we want to zoom in a little bit more on our chromosomes, trying to look for a little bit smaller pieces of our genetic information that maybe is missing or extra, and to kind of see where it is on what specific chromosome as well. And then if we want to detect what we call sequence variants, we need to zoom in even further to look at the individual letters of our genes. And this is done through gene sequencing. A lot of times panels are used in terms of sequencing where they would look at, multiple genes. So 5, 10, 100 genes at a time with sequencing. A lot of times the gene panels are geared or moved together to look at genes associated with a certain health condition, so like seizures or autism. And then there's also exome sequencing, and this is used to look at variants in all parts of our genes that provide our body with instruction. So these parts of our genes are called exons. And then we also have additional, which is another layer which I didn't add on, which is slowly coming more available. It's called genome sequencing, which is just looking at all of our genetic information in total. So our ability to detect variants is improving with, as we just saw, we went from karyotype looking at large copy number variants and some microarray sequencing and, exome and genome sequencing. But there's still a lot to do once we find a change. We need to understand what that change is doing. So today, the challenge is really in classifying the changes or the variants that are identified. When a genetic testing laboratory identifies a genetic variant, the next step is to classify that variant to determine if it impacts how the gene works in the body and if it could be associated with a health condition or a developmental difference. So we ask the community, the genetic community at the laboratories, ask a lot of questions when they find a specific variant. And they kind of say, has this variant been seen before? Do we know what this change does on the body? Has this been studied? Has this been found in anybody else? Has this been found in people that have similar symptoms or similar health history? And they take all this information together in order to determine what we call a variant classification. What is that impact of that variant on us, on our body? So likely pathogenic and pathogenic means that change is what we would call that variant that we see does impact our health. It may be responsible for a little bit of the clinical history that we are talking about or some of the symptoms that we are exhibiting. Benign and likely benign means that most likely this change has no impact. It is a normal change, and we all have these normal changes in our body. And then uncertain significance really puts us in this kind of borderline of we're not too sure. Maybe there's not enough evidence either showing that it does cause a health difference, or maybe there's not enough evidence to see that we have not seen it in enough people to know that it does not, or it's benign. So this is kind of what the laboratories and the doctors and the clinicians all have to kind of weed through when we start looking at genetic variants. More importantly, we also want to talk about how can we review these variants and review these changes and go through it in a very structured way and classify variants in a reproducible way, meaning everybody can hopefully get to the same conclusion with looking at the same pieces of evidence. And this is really where the clinical genome resource comes in. So the clinical genome resource, or ClinGen, is an NIH-funded project that is dedicated to building an authoritative resource that defines the clinical relevance of genes and variants for use in precision medicine and research, and ultimately hopes to understand what genes and variants are associated with disease and how genomic information can inform clinical care. There are a number of pieces of ClinGen that kind of you see in this graphic or summarized in this graphic. Today, we are going to really focus on this kind of top left corner, patients. So for some time, ClinGen has encouraged various stakeholders like laboratories and researchers and clinicians to share genomic and health data. ClinGen feels strongly that patients are also an incredible source of this additional information. Patients are experts about their own health and their health history, and they can provide much more detail about their symptoms over time, their diagnosis. ClinGen felt that access to this additional information could help increase our understanding of genes and genetic variants. So in 2014, ClinGen launched a patient registry called GenomeConnect. So GenomeConnect was launched to empower patients to share their de-identified genetic and health information with publicly available databases. So that's genetic information, and now, or genetic data. Sometimes it's referred to as genomic data. So just like to say that it's very similar. And now we are going to move over and discuss how can individuals participate in data sharing. And this is really focusing on GenomeConnect, which is ClinGen's patient registry. So GenomeConnect is open to anyone who has had genetic testing, regardless of results or diagnosis. It is not gene or disease specific. The platform consent documents are available in English and Spanish, and participants can enroll online. They consent to de-identified data sharing and recontact over time. They provide their health history through a number of surveys, and they upload a copy of their genetic test results. After each participant consents, each participant receives an invite to complete an initial health survey. And this initial health survey is used to capture kind of overall, how is this person's health? It asks a series of general questions across the 17 broad categories or of our body systems. So for example, it may say, do you have any issues with your heart? Do you have any issues with your eyes? And then if you answer yes, additional questions will populate. So ask broad questions, and then it moves on to a little bit more specific questions based on the responses. We also have additional surveys that can be assigned based on the participants' initial responses to the health survey. So if someone says, the question is, do you have any problems with your heart, such as extra heartbeats? Then we have a specific survey called arrhythmia, and that will kind of go into what are different questions are maybe associated more specifically to the arrhythmia symptoms that you are experiencing. Similar, if you say, I do have a family history or I do have a personal history of cancer, there's an additional survey that tries to ask more specific questions, getting a little bit more information about the cancer diagnoses that you may have experienced or that is in your family history. So why do we want to collect this information? Well, from our experience, we have noted that patients can contribute additional health information and other details about their history that can be quite valuable in increasing our understanding of what variants and genes cause disease. Laboratories and clinicians might have health details and family information, but they often do not include these details when they submit to these public databases, because most of the time researchers submit large amounts of data at one time. Patients that through So patients and working with patient registries, we can help improve or expand this information. So patients working with patient registries can share this additional health data. So that is how we collect someone's health data. GenomeConnect collects the genetic information or the genetic data through the uploaded report. So participants share their genetic testing results by uploading a PDF or an image of their results, or you can provide GenomeConnect with permission to request records from a laboratory or the doctor that may have ordered the test or performed the testing. And then genetic counselors on our team review that report to ensure that all the genomic data is collected in a standardized way and to ensure its accuracy. We pull all of this information, so all of the genetic and health information that the participants provide in our registry. We work through a process of de-identifying that information, and then we share it with ClinVar. So ClinVar is a publicly available database of genetic changes and their classifications. This database is where many clinical laboratories submit their variants. Many clinicians or researchers also use ClinVar when they're doing their work or they have questions, or they also may submit to ClinVar as well. And so submitting this information to ClinVar makes that information publicly accessible for all of these other stakeholders, like I just mentioned, to gain access and you're able to see this information. I'm sure a question is, what is ClinVar? So let's go through that. So ClinVar is a publicly available database, and it holds de-identified information about genetic variants and its relationship to human health. So ClinVar is public and it is freely available. It is a major resource for healthcare providers, laboratories, and researchers who are caring for patients, performing testing, and researching genetic conditions. The database is submission driven, meaning that it relies on all of us, on labs, on clinicians, and everyone else to share this data. So ClinVar is how we pull resources and knowledge in effort to advance our understanding of genetics and health. Here is a screenshot from ClinVar. So you can see up top has the gene, and then it has a specific information that helps everyone know what change we are looking at in that gene, specifically where it is located. It also gives the overall classification. So this gene is classified, this variant is currently classified as uncertain. And in the middle here, these are so many different ways of naming this same change. And so this is why when you upload a report, our genetic counselors really pull out that information in structured terms so we can make sure that we're all talking about the same exact change. This is another screenshot directly from ClinVar, and the yellow is what I want to kind of bring your attention to. This is a GenomeConnect submission. So the submitter here, the submitted column, all the way down, you can see GenomeConnect, ClinGen. This is a little bit of the information that comes up on ClinVar. So this is the participant information that has been de-identified and shared. So it doesn't include name, date of birth, but it does include some additional information that we can see, like the specific change. This lab information tells us which lab did the testing, what was the date that they did the testing, and what was the classification that the lab provided. On this expanded view, you can see it includes things like how was the classification provided by the laboratory, when it was reported, what are the details specific to the patient or the participant. So thinking ages of diagnosis, what was their sex assigned at birth, what symptoms did this individual provide through the completion of the health survey. And this can help other users of ClinVar, which we will get to in a few slides. And lastly, what I really like to showcase is thanks to our participants in GenomeConnect, we are also sharing novel information with ClinVar. About 18% of the time, information or genetic information that our participants share, when we put that up into ClinVar, we are the first individuals to submit that variant, that information. So if it wasn't If our patient participants didn't engage and didn't participate in GenomeConnect and they didn't upload those reports, then this information would not have been shared with the genetics community. And then 82% of the cases, we are providing those additional phenotype information, that a different clinical information and symptom information that everyone is providing when they complete those health surveys. And I apologize that my 2024 got caught off. The data is of about 2 weeks ago when I initially put it all together. Okay, so that is how individuals can share and participate in data sharing. And now kind of the meat and potatoes is, why is this so important? Why is data sharing important? Why do we encourage everyone to engage in data sharing? Why does Clinton put so much effort into getting laboratories and patients and researchers and clinicians all to get involved in this data sharing? So we know that pulling resources and knowledge is important to advancing our understanding of genes and health. So this additional information provided by the participants can help with variant classifications. We refer to this information as case level data. I'm sorry for that. So case level data can help testing laboratories first understand, one, what is this gene? What is this gene doing? Is this gene associated with diseases? If we see a change in this gene, what is it actually doing to the body? What is its impact? If the gene has not been associated with disease, then it is really hard to understand different variants in that gene. So if we don't know what the gene does, then it doesn't matter what changes we see. We have no idea if that change is going to have any impact on an individual's body because we don't know what the gene does in the first place. So this kind of goes back to gaining better understanding about genes and genetic variants and getting more and more information about how genetic variants may be associated with certain health symptoms. So that is one thing. We're getting more information, and that can help us understand all of the genes in our body. We have 25,000 genes. So it is really good to understand and learn a little bit more about what each of those genes does. Additionally, even if we understand what that gene does, having this additional information that participants are able to provide, we might be able to get more information, expand what we know about this genetic variant and how it impacts health. We might be able to get a better understanding of what certain genetic variants can cause certain symptoms. What are the symptoms of the condition? What can we do to best care for individuals with this diagnosis? Again, the participants sharing this information is really helpful in answering these questions. So questions like, how often are our symptoms seen? At what age are symptoms seen? What is most likely to be expected when someone is age 7? Maybe what is most likely to be expected when someone is age 20 or 70 or maybe even 110, right? So having all of this additional information collected throughout the whole lifespan of a person is really helpful and provides a lot of great data for everyone working to best care for these individuals and having the best idea of what treatments may be beneficial. So ultimately, data sharing is important because it can help increase our understanding of genetic conditions and can help doctors and genetic labs better understand genetic changes that cause disease. This increased understanding can help identify interventions and treatments and can help individuals get a clearer genetic test results. Our hope is that the more information collected, the better we will understand how genes affect health and ultimately how to best provide care for patients. Okay, I know that I have talked a lot, so thank you all. Now I'm going to go through a few examples which I think really help showcase the benefits of data sharing and really help showcase the importance of individuals being involved in data sharing. So we had a family enroll in GenomeConnect. The family shared their child's genetic and health information with GenomeConnect. The genetic report identified 2 changes in the EMC1 gene. EMC1 is associated with cerebral atrophy, visual impairment, and motor delay. The report that they shared indicated the individual inherited one EMC variant from their biological father and the other variant from their biological mother. And both of these variants were classified as likely pathogenic. This information most likely explained the symptoms that this individual was exhibiting, and this family most likely had a diagnosis for some of the health concerns that they identified in their child. But they were still really eager to participate in the community, engage in data sharing, and so they were it was really great of them to enroll and to share this information. We were then able to, as we go through, collect that health and genomic information that they submitted. We submitted it to ClinVar, and we were able to share that this variant, this EMC variant, was likely pathogenic, and we were able to include some of that phenotypic information. When we were reviewing other ClinVar submissions, the GenomeConnect team identified laboratories that classified this specific EMC1 change as uncertain. And so this is a conflicting variant classification. We have that it's likely pathogenic, and this other lab has that it was uncertain. So GenomeConnect, we said, okay, let's reach out to this lab. Let's meet, let's discuss. How come we have differences in variant classification? Is there any additional information that we have or additional data that we have in our patient registry that may be able to help this lab be able to provide a little bit more information to that lab? And so we did. We reached out to the laboratory and we were able to say, hey, this participant, shared their genetic report. They were identified to have two, what we would call co-occurring EMC1 variants. They were on that they were inherited from the individual's biological parents. And these changes that we identified in the individual also kind of is consistent with a little bit of the symptoms that they provided. And so we were able to say, is this information useful to you as a laboratory? Does this help with the data that you have? Can we reclassify? Can we reevaluate? Can we relook at your classification of the same EMC1 variant? The laboratory said, this is great. Thank you for sharing. They were able to use that extra information that we provided when we reached out to them. They did their own internal reclassification and reevaluation of that variant, and then they were able to reclassify that variant, the same EMC variant, as pathogenic. Now, this allows the laboratory to reissue amended reports to other families and other individuals that were tested with them. So thanks to this one family that shared their genetic and health information, we were able to share this with the community, with another laboratory, and then that laboratory was then able to share this information, this updated classification, with a number of other family members as well. So this case is an example of a pilot project initiated by one of our genetic counselors on the GenomeConnect team, and we are really hoping to expand this work and continue to demonstrate, and as we continue to demonstrate the importance of engaging participants in data sharing. Okay, last but kind of a really important question. What are the benefits of data sharing for the individual participants? So what are the benefits of you engaging in data sharing? Are there any benefits? And there are some. And so that's just part of the reason why we also wanted to start GenomeConnect, in order to provide some of these benefits to the individuals that directly enroll in GenomeConnect. So one, oh, I'm sorry that error on my part, that did not adjust the font. So through the registry, we can facilitate genetic result updates. So we can recontact families about potential variant updates. And this is something that we know is very valuable. At this time, there's no real consensus on how best to do it. There are often some barriers that laboratories and clinicians experience with recontacting families over time. One is kind of resources and availability of terms of staff and if we have all the updated contact information for the families. So the registry can offer one more additional way for families to remain informed about their results. So in particular, through our data sharing, we can identify instances where participants' report maybe is out of date compared to the current understanding. So maybe in ClinVar, we see that variant is classified as likely pathogenic by the laboratory that did their testing, but when the participant had testing years ago, it was listed as a VUS or a variant of uncertain significance. So at that time when the participant was tested, we just didn't have enough information about the specific variant. There wasn't enough data available or enough case studies available in order to provide a more definitive classification. But over time, we got more information, there was more learned about it, and the laboratory was able to reclassify that variant as pathogenic. And then this is something that we can share back with our participants that opt to receive such information. And to date, we have shared over 250 updates with our participants. This graph just shows kind of all the different various updates that we have seen and we have shared back. So we have seen some variants that were classified as a variant of uncertain significance and that maybe was reclassified as likely benign or likely pathogenic or pathogenic. And then some variants that were classified originally as likely pathogenic and maybe moved up to fully pathogenic. So we have seen a number of different various updates that we have been able to share back with our participants over the life of GenomeConnect. So just to recap, how do we do this? Genetic and health information is shared by GenomeConnect participants. That information, we extract their health information from their surveys, we extract the report information or the genetic information from their uploaded reports. We are submitting that all to ClinVar. We then review the ClinVar information and we look for any variant reclassifications. If we have identified a variant reclassification or a change between the classification of our GenomeConnect participants and the reporting laboratory, we reach out to the reporting laboratory. Many times the reporting laboratory has already issued an amended report, and we are just another way or another resource of letting that individual and that family know that, hey, there might be an update. We would encourage you to reach out to your healthcare provider that ordered the testing and request an updated report. So this is a feature that all participants are able to opt into or opt out of. And this is available when you set up your GenomeConnect account or if somebody wants to go back and edit these features of their GenomeConnect account. So this is an example of something that we have also experienced, kind of that same report update. We have seen that a patient who enrolled in GenomeConnect, they shared their reports. They had testing way back into 2005. At that time, they were identified to have a variant of uncertain significance in the HECW2 gene. This participant enrolled in GenomeConnect. We extracted that genetic information. We submitted this de-identified data to ClinVar. When we were reviewing the information from ClinVar, we saw that this specific variant, this specific HECW2 variant had been reclassified as pathogenic or disease causing. And we said, okay, we see that the lab now in 2016, at the time when we did this, at 2016 does classify this as pathogenic. We reached out to the laboratory. The laboratory did confirm that yes, it did change its classification. It is now classified as pathogenic. They would reissue a report to the ordering provider. We reached out to our participants and said, hey, there might be an update to your results. We would encourage you to contact your ordering provider or contact the laboratory that did the testing. And so then they were able to get a little bit more information. So we like to give this option to patients that enroll in GenomeConnect, and we like to see if they are interested in getting these updates. And so these updates are all sent out via email And so then there is some information in the email that the participant can take to their provider as well. Okay, another benefit of GenomeConnect is that GenomeConnect enables connections with researchers or clinicians interested, maybe interested in studying a specific gene or a specific variant. So if a researcher is interested in a gene and reaches out to GenomeConnect, we are able to recontact the participant to collect additional information about their health history or connect them with additional researchers. At GenomeConnect, we do not directly hand out or give your contact information to any researchers or clinicians. We will send our participants an email saying, hey, this researcher is interested in working with individuals that have variants in the specific gene. Here is this researcher's contact information. Take a look at the study that they're proposing. You all can decide if you would like to reach out to that researcher directly. The same goes if there is a research study. Again, we don't send anyone's information directly to the research study. An email will come from GenomeConnect, and we will say, based on the information that we're able to pull out from the genetic report, you may be eligible to participate in additional research opportunity. And here is the information about that research opportunity. Take a look at it, discuss it with your family. Do you think that this is a good opportunity or a good option for you? And then you can reach out to that study directly. And another benefit that we have is our participants can match with one another. And so participants are able to, again, this is a feature that you select. Do you want to be connected with other people in the registry? You can say yes or no. And then if you say yes, then individuals that maybe have a similar gene, a similar diagnosis, or a similar geographic location as you would be able to connect. And then you would get again a message coming from GenomeConnect saying, hey, this family, this participant matched with you. They would like to connect. Do you want to email them? Do you want to set up a time to speak with them? So again, we like to provide all these options, and these are all different features that a participant can consent to or consent out of. And now I have completed my talking at you, and now it's the time for questions and answers. So I am going to stop sharing my screen so I can see if any questions come up.
Thanks, Caitlin. I think one question that came up while you were talking was sort of where to go if you have questions about your own genetic test results. So if you could maybe talk a little bit about that, that would be lovely.
Yeah, so if you have questions about your own testing result, that is a great question. Those are definitely challenging results to read. One thing that we do encourage is maybe contacting the provider who did the genetic testing. There are also some resources to meet with a local genetics provider. I'm not sure if we should share, we can put those in the chat, and that's able to, oh, perfect, thank you, Jules. And then you are able to contact a local genetics provider, and they can really walk through the report kind of step by step, going through what this information would mean for you. What does it mean in terms of your health history? What it means in terms of your family history? You can also look up that specific change in ClinVar, and that maybe provide you with a little bit of information of what other laboratories or what other researchers are saying about that variant. Do they have any additional data that they have posted on their ClinVar submission? And lastly, we also would encourage everyone to go to ClinGen, which is, or clinicalgenome.org, and that is our website. And we do have researchers all the time looking into the genes, looking into variants, trying to determine which ones are associated with health and which ones are associated with what type of disease, what are their features. And so they have a lot of information in terms of what is this gene doing what can it cause, what health conditions does it associated with. And so that goes into a big category called gene disease validity. And then we also have a lot of researchers looking at specific variants in those genes to determine what ones do really have impacts on individuals' health.
Thanks for that, Caitlin. Feel free to put other questions in the Q&A box while we wait to see if there are others from those in attendance. Caitlin, I think one other question we get quite often is when? Like, how long will it take to get an update, or will I definitely get an update? So could you maybe talk a little bit about that piece of things?
Yeah, that's a great question, and that is a difficult question to answer. The when is kind of unknown. Not everyone does get an update, and that really is unfortunate. But as more and more individuals share their data, and as more and more information, or more and more laboratories and researchers and clinicians participate in data sharing, that can really help everyone as a community obtain more definitive answers when they do genetic testing. So that timeline is a little bit uncertain. It really depends on kind of everybody else sharing this information, collecting that data. And so we appreciate everyone, and thank you so much for joining and for sharing that information. It is super helpful. The one question, unfortunately, can't put a time frame on it, but if we do learn of an update, we definitely do reach out, and we definitely like to share that information with all of our participants.
Thanks, Caitlin. And I think that a similar question is, what information does it take to upgrade a variant? And perhaps that's a similar story of it really depends, right?
It really depends. There are lots of different kind of characteristics or guidance that we have in terms of when we evaluate variant classification. So we look at population allele frequency. So how common is that variant? How rare is that variant? It really also, we do take a lot of case level data, like we said, from participants sharing their health history. And so that also information needs to be shared in the literature for us to have enough information to say these change, we see these symptoms, and we see it consistently over time in a number of individuals. So there are a lot of different factors. I know a lot of laboratories are, thankful that they share their information with ClinVar, but as more and more laboratories do the testing, it is really great if everyone can get on board with sharing it so we can all move forward together.
And one of our participants asked if there's any way for them as patients to report that their variant is disease causing and their medical files show the hallmarks of the genetic disease. So talk a little bit about maybe, how we share data, what we can share, if that might be helpful.
Yeah, so that's really great information. So the information that we share, when we do submit it to ClinVar, we share the information directly as reported by the laboratory that did the testing. So we will take the classification that the laboratory has put on their report, but we will also include those additional health information that the participant provides through their survey. So maybe if that's probably most likely what's also being seen in their health record, those symptoms and those features, and then we share that information. And so that then goes to anybody else or any other group that is evaluating that change in the future. There is now, hey, we have this as well. We have this level of case data as well as what we had before. So sometimes, it takes a few of these cases to end up tipping it to a more definitive state, but those pieces of information is really great because a lot of times we are able to share that additional phenotypic information that the laboratories are not able to provide when they share their information in ClinVar. The laboratories are really saying, we've seen this variant maybe 5 times, 6 times, and they're just putting that variant information, and they're not really including what we call that case level data, all that health information that's directly the patient is providing.
Thanks for that, Caitlin. And I'll highlight, Caitlin told that story of the EMC1 variant, and that was reclassified really because we were able to share that a different variant was detected in that participant. But we also have cases where having those additional details, sending those to labs is really prompting updates. So we have some examples where that's been really helpful just to tie that up too. Thanks, Caitlin.
Yeah, thank you. These are all great questions. You guys are amazing.
I think that's all of the questions I see so far. I'm going to give it one more moment if that's okay. But maybe we can remind folks about our upcoming webinar, see if there are any last lingering questions.
Absolutely. Okay, so these are upcoming webinars. I should have removed the first one, sorry. The next one is on April 25th. That will be at 7 p.m. Eastern time as well. And this will be talking about how to read a genetic testing report. So we'll walk through a little bit of a mock genetic testing report, looking at some of those key pieces of information that is really useful to kind of reading your report, seeing where the information, what information is most valuable. And this will be on DNA Day. So April 25th is DNA Day. And then on June 25th at 3 p.m., we are going to have a speaker come in, or two speakers actually, and they're going to talk about the Brain Gene Registry, which is a registry that the GenomeConnect partners with. And this is kind of going a little bit on how I mentioned how we work with other researchers, and we sometimes tell our participants about other research studies. And this is a research study that we have been able to tell our participants about, and this is a research study that we work very closely with. So it'll be really exciting to learn a bit more about it. And last is just a massive thank you for all of you for joining today. Thank you for taking the time out. Thank you for registering. Thank you for all of your questions. And thank you to all everyone for just engaging in this awesome feat of learning more about our genetics and health. And if you have any questions that we didn't go through or that pop up in your head, you can email us at info@genomeconnect.org. And I know I said it like 100 times before, the webinar will also be on our YouTube playlist, so it will be available as well.
Thanks all. I hope you have a wonderful Rare Disease Day tomorrow. Thanks, Caitlin, for walking us through that. We hope to see you all in April.
Have a great night. Thank you.